This is Audiommunity, a podcast about our body's never-ending fight with the outside world. Today, we're talking about the autoimmune disease multiple sclerosis.
And so what you're really talking about when you're talking about multiple sclerosis is the inappropriate infiltration of the immune system into the brain. And when it does infiltrate the brain, it causes this really destructive inflammation. And because the brain is so sensitive to that sort of attack, you get very long-term debilitating disease.
And we're talking about how our immune cells use the bloodstream and lymphatics as highways to move around the body and get to the tissues they need to be in. Basically, imagine trying to take an off-ramp from a freeway when you're going like 500 miles an hour. It's just not going to happen.
Or it could,
T-cells.
and it would be catastrophic.
Right. This episode was recorded over a month ago, and a few major life events intervened and prevented us from getting it to you sooner. But now that's done, and we should be back on track with our bi-weekly schedule going forward, and we hope this episode was worth the wait. Hi, welcome to Audiommunity. I'm Kevin Bonham. Joining me today is Matt Woodruff.
Hey, everybody.
And unfortunately, Kate couldn't join us today, but we are going to press on without her. Hopefully she'll join us next time. Today, we are going to be talking about a number of different things. We're going to talk about some autoimmune disorders. Specifically, we're going to talk about multiple sclerosis, which is a really, really hard thing to say. So mostly we're just going to refer to it as MS. But that is an autoimmune disorder that attacks the brain, and specifically the myelin sheaths that surround the brain. And we're going to be talking a lot about what those things are in a little bit. We're also going to be talking about the ability of the cells of our immune system to move around in the body, to migrate between different tissues, between the blood and the tissues, and how that ability affects the progression of MS and how we treat it. So first, I'm going to give you a quick introduction to the idea of autoimmunity, and we're probably going to spend a lot of time in coming episodes on autoimmunity, but because we're talking about MS today, I want to just give a brief introduction. So autoimmunity generally is when your immune system overreacts and attacks your own cells and your own tissues. And this can happen for a number of different reasons in a number of different areas of the body. You may have heard of lupus, probably heard of MS. It's a little bit different than something like an allergy, which is an inappropriate immune response to something from the outside world. Autoimmunity is an inappropriate reaction to your own body. And one of the main problems with this is that you can't just get rid of your own body. You can't remove the thing that your immune system is attacking. And so your immune system just keeps getting activated.
Did you ever watch House?
Yes, I did.
I loved the recurring...
It was a guilty pleasure.
Yeah, the recurring lupus joke in House. Yeah. I thought it was hilarious. And it was before I actually knew what lupus was, really. Did you see the episode where they finally were like...
Diagnose a patient with lupus?
Yeah, or someone says, it's never lupus.
Right.
Yep. Anyway, that made me super happy at the time.
Yeah, but the reason that happens is, or the reason that lupus is often a potential diagnosis is because lupus, in particular, is basically antibodies against everything.
Yeah.
It starts often with antibodies against your DNA, but it can basically spread throughout the body, and it just has a lot of different potential symptoms, very wide constellation of symptoms.
And it's not even clear. It's not even clear what starts it, right? The first steps in lupus aren't even well known. There's a lot of indication that perhaps it's inappropriate cell debris clearance. There are a lot of things that could be happening, and really the only thing that we know is that this huge spectrum of disease, which is characterized, I guess, by these autoantibodies. We don't know what to do with them. It's probably actually multiple different diseases all under one diagnosis. But anyway, that's lupus.
Yeah, it's actually interesting, too, because what you just said, that we kind of don't know how it starts. It's what we call the etiology of the disease, the beginnings of it. That's sort of true for all autoimmune disorders. There's a lot of immunology that's been done over the last several decades trying to figure out where the T cells come from that start autoimmunity, how the initial immune reaction starts. We talked a little bit last week about how an immune response can't start unless you have these pathogen-associated molecular patterns that come from microbes. But most autoimmune disorders don't seem to be associated with any particular infection. There are ways of deleting autoreactive cells like B cells and T cells, and there's a whole host of different ways that the immune system tries to prevent autoimmunity, but it still happens. And we've gotten really good at understanding a lot of the ways that the immune system protects us from autoimmunity, but that still leaves open the question of, well, if we have all of these mechanisms, why do we still get autoimmunity? But yeah, so getting back to MS, this is an autoimmune disorder that specifically attacks the brain. So unlike lupus, which has sort of systemic consequences, can be attacking all kinds of different tissues, MS is specifically targeting the myelin sheath around the neurons of your brain.
Yeah, and so actually the name multiple sclerosis describes what's happening in the brain at the time of the disease. The name of the disease is actually pretty indicative of the disease itself. And so if you take brain scans of people that are developing multiple sclerosis, what you'll find are these white lesions that sort of pop up and then they go away. But when they go away, they sort of leave this scar behind, right? And that's where the term sclerae comes from. It's just a scar on the brain, essentially. And so what you're really talking about when you're talking about multiple sclerosis is the infiltration of the immune system, the inappropriate infiltration of the immune system into the brain. And when it does infiltrate the brain, it causes this really destructive inflammation. The brain isn't well equipped to handle that information. And because the brain is so sensitive to that sort of attack, you get very long-term debilitating disease. But I guess to really go into MS, we should provide a small overview maybe of how the brain works, how the nervous system generally functions. And so when I think about the nervous system and how it develops, I actually think of strawberries that I used to grow with my parents growing up in the backyard. And the reason I think of strawberries is because one of the coolest things about a strawberry plant to me at the time, and I didn't understand how plants really reproduce, is that they send out these cool runners, right? So you can have the plant and it grows, and then at some point that plant shoots out a runner and that establishes a new plant. So if you're thinking about the nervous system directly, you have all of these different neurons and they're all inhabiting roughly the same regions of the brain, although it is highly organized. So I think a neurologist would take me to task for that. But there are highly organized
This is not This
cells.
Week in Neurology.
Right, exactly. So they are highly organized, but at the beginning, they are sort of making these random connections. The cells are there, and they're sending out these runners, and they're making contact with other cells, right? So in the end, you have this huge mess of a network of all of these interconnected neurons, and they're all talking to each other. And the reason neurons have to talk to each other is because really, when we're talking about nervous system firing, we're actually talking about electrical currents, right? We're talking about the movement of an electrical potential from one side of a cell to the other side of the cell and then into a new cell, who will then transfer that electrical current, which means that the two cells have to actually be touching each other. And so I don't know, maybe circuitry is a good example of this, like how a processor works, if anybody is familiar with that.
Except that no engineer would design an electrical circuit the way that our brain is wired together, because you constantly would have crossed wires. You'd have all of these things where one thing is sending out electrical potential and it's getting interfered with by other wires that are jumbled together.
Right. So you essentially start with this jumbled mess of crossed wires, and everybody's talking to everybody else, albeit sometimes inappropriately. But the important part of that is some of those connections are going to be really critical in the downstream function of your nervous system, right? So let's say you're talking about the blink function, right? In order for you to blink, your brain has to send out the blink signal. It has to go through your neurons. It has to arrive at the muscles that control your blink response, and then that muscle needs to fire. So you can imagine there is a linear signal, a linear electrical signal that goes from one part of your brain out to your eyelid, and then probably some feedback saying, yep, I blinked, everything's cool, right? So along that pathway, every single connection is important. But the other mess of connections that aren't important are sort of interfering with that signaling when you're first developing. And so this is, in my understanding, sort of how the nervous system develops, where you have connections that are constantly being tested, and the connections that are functional are strengthened, actually. They get more robust. Those cells decide, yes, I do need to talk to my neighbor. And the sort of superfluous outside, crossed wires stuff, all of that actually gets pruned away. And so what you end up with is you have these neurons, and only the connections that are actually helping you as a person and an organism function, only those connections remain. And it's something that people don't really think about, but there's a really good reason that you don't have memories from when you're 1 or 2 or even 3 years old. And the reason for that is when you're first born, all of your neurons still have all of these crossed wires. So your brain is still trying to figure itself out. It's still trying to test all of those connections and exactly figure out which ones are important, which ones aren't. And so there's this huge remodeling when you go from about, I think it's 3, Kevin, is that about?
I am also not a neuroscientist.
Okay.
Or a developmental
I'm fairly
psychologist.
I'm fairly certain it's around 3, but anyway, there's a huge pruning that happens between the ages of 1 and 5, let's say, to cast a really wide net so I don't get beat up in lab on Monday. There's this pretty big range, but there's a huge amount of pruning that happens, and one of the reasons that you don't form these really lasting memories early on in life is because all of these synapses are constantly in flux and they're constantly being pruned. But by the time you're about 5, 6, 7, 8, a lot of those neurons have really decided what's important. They've strengthened the connections to other cells that will help you later on in life, and they're set. A lot of those things won't change as much as other cells in your body will change. And so in addition to this really intense pruning process that's happening and making sure that cells are contacting each other in a way that is productive for the organism and, basically developing into a fully functional nervous system, it's also important to take a look at the really small level physical components of the neuron, because this is where we'll eventually be getting to when we talk about what's going wrong in MS. So you do have these connections, but when you're generating those connections, like I said, with the strawberry plant, you've got these really long runners. And those long runners is what is actually going to relay the electrical action potential from the cell body all the way down the length of the cell to the next cell. Right, so these can be huge distances. I mean, we're talking feet in some cases, where you have a cell body of a neuron in your peripheral nervous system, and then there's an axon the length of your arm, say, and then finally whatever it's actually talking to. So
yeah, I think some of the early neuroscientists, they were studying like this certain species of squid that had just an enormous neuron that was feet long and a few millimeters wide, and that sort of allowed them to do a lot of the early neuroscience experiments. But it's not unique to squid, and I'm actually going to look that up right now.
Sure. Well, we'll get confirmation on that. But regardless, they can be really, really long sometimes. And so you can imagine it's not easy to transmit that sort of electrical potential over such huge distances. What you have in order to sort of get around that length problem is this sheath that encompasses, it almost wraps up the length of that neuronal process. And we call that sheath myelin. And so myelin, Kevin and I actually were just talking about my misconceptions of myelin. For a long time, I thought it was just a direct insulator, like copper wire is insulated. But it turns out that the function of myelin and that sort of wrapped cover actually speeds up the transfer at which this electrical signal is passed down the length of the cell. And so what I mentioned before was that these cells are really dependent on interacting with each other, and they get feedback on which connections are productive. And so if you start to lose the myelin sheath, it means that you can't transmit the signals quite so well, which means that you're not going to get the same sorts of feedback saying, yep, this was a really good connection, it's working appropriately, right? Something is going wrong. And so what ends up happening is the neuron will actually wither and die. And so by inhibiting your ability to effectively transmit these signals, you basically get inappropriate pruning of your neurons, which ultimately, going back to the case of, the eye blink response, let's say all of that myelin, specifically in your eye blink response, was degraded. What that means is that those neurons would slowly retract, they would stop signaling to each other, and at some point your brain would try to fire the blink signal, but the electrical signals that get it from your brain into the muscle in your eyelid, they just won't work. So this is sort of my rudimentary, I guess, understanding of the neuronal system, but I think it'll work in our case where we're talking about MS as a disease. And I mentioned earlier, or Kevin mentioned, someone mentioned that MS is the inappropriate infiltration of the central nervous system by your immune system. And what really happens when that infiltration occurs is that at these lesions, these sclerae, basically you get local amounts of really potent inflammation, which leads to the local demyelination of the nerves in the surrounding tissue. So that entire region that the immune system is acting on starts to get inflamed, it starts to lose its myelin, and so as a result, it starts to signal less effectively. And so all of the downstream symptoms associated with MS are basically a result of this slow degradation at these sites of immune activation and infiltration.
Okay. But by the way, just as an aside,
Yeah.
We actually have an axon called the squid giant axon, and it is a very large axon that controls escape behaviors.
Did we define axon? I don't know that we defined axon.
I don't think we did. But I just want to point out that I was right about the squid.
Okay.
And apparently Hodgkin, I'm guessing of Hodgkin's lymphoma fame, figured out how nerve impulses are generated using squid axons. An axon is the part of a neuron that is transmitting signal. So you can think of neurons basically with three general features. There's a cell body that has all of the things that make a normal cell function, metabolism and things like that. You have the dendrites, which are where neurons receive information from. This is not to be confused with the dendrites of a dendritic cell, which is an immune thing. Neuronal dendrites are just extensions that are looking for signals, and then usually a single, they might have many dendrites coming in, but usually neurons have a single axon coming out, and an axon is where information is transmitted away from the cell.
Right, and in the strawberry plant example, I guess I can't really distinguish between the dendrites and the cell body, but the axon itself would be the runner. Maybe leaves, would that? That doesn't really count.
Here's the problem. Break down,
here's the problem is that strawberry plants don't talk to each other in the way that neurons do, but that would be a cool paper if they did.
Yeah.
In any case,
Plant communication is super cool, and it's mostly with volatile organic compounds. And we could talk about, no, it's not immunology, so not appropriate.
Maybe we'll break it someday.
There actually is communication between plants as a defense against parasites, like viruses, bacteria, and mostly insects, actually. But plants can actually emit chemical signals that warn nearby plants that there are things that are attacking.
Sometimes my bananas warn my other bananas that one of them is ripe, and then the rest of them are warned appropriately. And then I have rotten bananas, and then I have banana bread. So it really all works out in the end, I guess.
Speaking of the Cavendishes,
can we just go back to them at every opportunity? That
was my plan. Yeah, I actually just heard a story talking about this disease that is raging through banana plants, and apparently we originally adopted the Cavendish cultivar of bananas because there was this other, I think it's called Racer. I'm gonna actually look this up too.
Banana diseases. What does Wikipedia say about banana diseases?
I just searched banana race and I'd see YouTube videos of people dressed in banana suits. Race 1, yeah. So
yeah, thought
actually
to be
the reason that
to Panama disease tropical race 1. Yep.
Yeah, so the reason we originally picked the Cavendish cultivar of bananas is because it was resistant to this infection called Race 1 that was devastating the previous cultivar.
Which is, by the way, the Big Mike banana.
Nice.
Yep.
And now there's a new viral infection called Race 3 that is actually affects Cavendish bananas as well, and it is starting to move. It used to be, this story that I just heard, it used to be only in, I think, Southeast Asia.
Well, in any case, we
should talk about banana diseases because we have a thing for Cavendishes.
Yeah, now. I'm for it. I like bananas too, so
that's
there's
true.
A coalition of interest. Yeah, so maybe if we try to move back to MS here, obviously the other component in MS is it's not just the demyelination and the immune response going on in the brain, it's really how the immune response starts in the brain, and really how specifically T cells find their way into the brain through something that we like to call the blood-brain barrier.
Right, and so the blood-brain barrier is establishing a perimeter around the nervous system. And it's not just the brain, despite what the name says. It's actually a sort of zone of what we call immune privilege. So the immune system doesn't typically get into the brain under normal circumstances. And that's because most of what the immune system does is cause inflammation. And if you get inflammation in your skin, if you get inflammation in your kidney, in your gut, all of these tissues are able to repair themselves. And so, let me start this over. All of these places are places that infectious diseases can get to. And when the immune system responds, it causes inflammation. Usually inflammation causes some tissue damage, but that's okay because once the infection is cleared, you can then repair the damage. The problem with inflammation in the brain is that repairing damage in the brain is much, much more difficult. And that is because of what Matt was talking about during development. The way that the brain knows where to send signals is a function of how the brain developed in the first place. And the signals that were deemed to be important during the development of the brain were the ones that were strengthened. And the ones that weren't important were the ones that were pruned away. If you have inflammation that damages neurons after development has already occurred, there's no way for the brain to sort of figure out what connections were important, how to send those axons to the right place. You'd have to sort of start brain development from scratch
in order
to repair those signals.
And this is also why when someone suffers, catastrophic brain injury, we see a lot of wounded veterans coming back from the war and things like this. The reason that it's so hard to regain those functions is because that's exactly what they're trying to do, right? The brain has been heavily disrupted. You have maybe some neurons that survived, but none of the connections that originally existed, even if you regenerated every single neuron that was there to begin with, none of those connections exist. And so if you're relearning how to walk, relearning how to talk, those sorts of things, you are quite literally forcing your brain to redevelop, which is why it takes so much time and why it's so hard.
And actually, when occasionally it is possible to relearn some of these things, often what is happening is you are training your brain to use neurons that are already there for a different purpose than what they originally had. You actually don't make a ton of new connections even after a really terrible injury. And there is some repair of the brain that happens slowly, but for the most part, when things are damaged in the brain, you can't get them back.
And this isn't just the brain. It's there are a couple immune privileged sites, and they're privileged for different reasons, actually. Sometimes the eye is immune privileged. It turns out that it's really bad for you to not have an eye. Sight is a really, really good evolutionary driver, so it's important for your immune system not to mess up the really nice clear globe that makes up your eye. Also, things like testicles. You don't want to have mutation offspring, and as a result of that, you don't want your immune system facing chronic inflammation and passing potentially damaged DNA onto your offspring.
Right. So,
now that we've discussed testicles and the immune system,
always happy to bring testicles into the conversation. So, the blood-brain barrier is a barrier that separates your blood from your brain and the rest of your nervous system. So before we get to the problems in MS with T cells going across the blood-brain barrier when they shouldn't, we need to talk a little bit about how the immune system gets around in the body in the first place. So the immune system is sort of weird as an organ in that it is not a solid organ the way a kidney just hangs out in one place, or your heart just has to stay in your chest cavity. If it moves, there's a problem.
Just has to hang out. Lazy heart.
Yeah, exactly. The cells of the immune system have to go everywhere. We've talked a little bit about the development of the immune system in previous episodes, but to sort of recap, in an adult, all of the cells that will eventually be part of your immune system start in the bone marrow as what are called hematopoietic stem cells. And these stem cells develop into all the different cellular lineages in the immune system, from T cells to B cells to macrophages. All of these cells that we've talked about in previous episodes, they all start as hematopoietic stem cells. As they develop, they go to different places. T cells go to the thymus for further development. We've mentioned before positive and negative selection of T cells briefly. We're going to get back to that in much more detail in the future. B cells develop mostly in the bone marrow. Other cells go into the blood and then to tissues where they end their differentiation. But in all of these cases, they have to move around. They move around mostly using the blood and another set of vessels that most people don't think a lot about that are called the lymphatics. And the lymphatics are a series of tubes, kind of like the internet.
I was really hoping you would mention that.
That drain the fluid from your tissues. So your blood contains a lot of fluid. Some of that fluid tends to leak out of the blood and into places like the skin, into your intestine. There's a lot of places that fluid goes, and it all needs to get back into the blood at some point.
Right. If you've ever sat on a plane for a really, really long time and you felt like your ankles were swelling by the time you stood up, the reason that you feel like your ankles were swelling was because they were. And what was actually happening there was blood was leaking from your blood vessels out into the surrounding tissue in your legs, but because you weren't moving, that fluid basically just sort of builds up in the tissue. It doesn't really have anywhere to go and sort of just collects. And as soon as you stand up and you start moving again, all of that fluid starts to get kicked out of your legs, everything returns to normal, and you sort of go about your way.
Yeah, this is also why patients that are on bed rest in hospitals have to be sort of periodically massaged by nurses to move the lymph around through the lymphatics. And that's because the lymphatics don't have a heart. They don't have a pump that is moving the lymphatics around. Basically, all of that fluid is being moved through the action of muscles. Your skeletal muscles, as you're sort of moving your arms, your legs, that is actually providing physical pressure that pushes lymph through the lymphatics past lymph nodes, which we've also mentioned, and eventually it goes to this thing called the thoracic duct that's in your chest, and lymph sort of dumps back into the blood at the thoracic duct.
You can almost think of it like a sewer system where you've got basically all of the excess flow dumps into these channels. So
right. And not to mix metaphors too much, but
I love mixing metaphors. It's my favorite kind of metaphor.
But the cells of the immune system use the lymphatics and the blood as a sort of highway to move around the body. So your lymphatics drain every tissue, your blood is obviously pumped through every tissue, and for a cell that needs to interrogate your entire body for the potential presence of infection, using the lymphatics and the blood as a way to get around is pretty convenient. And actually, T cells don't run around the body looking for infection. Usually what they do is they hang out in lymph nodes, which are, I think Matt described them in a previous episode. They're basically little balls of immune cells that drain the fluid from the tissue. And during an infection, cells like dendritic cells can actually bring pieces of infection to the lymph node to display to T cells. Sometimes pieces of pathogens or pathogens themselves, like viruses, can actually flow through the lymphatics into lymph nodes, and T cells can be alerted there. So the lymphatics and the lymph nodes are basically places where T cells and B cells can interrogate fluid from the rest of the body, looking for the presence of infection. But they don't hang out in a single lymph node. They move between lymph nodes because B cells and T cells don't know a priori where the thing that they're specific for is going to come from.
Right. Yeah. So they just jump around. People immunologists especially, like to talk about these things as they call them antigen libraries, a place where all of the antigen in the body is collected. Right, which is, it's, I guess, sort of true. But the T cells are just going to jump around B cells and, go from library to library until they see something that they recognize, and then they'll respond.
Right. And so as these T cells and B cells are moving around, they're not just flowing with the current. They're not just moving willy-nilly around the body, sort of flying around through Brownian motion, bumping into things.
Willy-nilly Brownian motion. I like it.
Yes, I'm not going to further comment on that.
All right.
Anyway, so as T cells and B cells are moving around, they are turning on and turning off different molecules on their surface that we call adhesion molecules. And these adhesion molecules allow them to stick to certain tissues. They're also expressing things called chemokine receptors that bind to chemokines, as their name implies, and chemokines are just chemical messengers that direct motion. There's nothing particularly special about the molecule. Chemokine is a general name for a chemical signal that directs the motion of cells, specifically cells of the immune system.
Right, and we call that...
Because again, your heart isn't moving much.
Right, we call that chemotaxis, which is where the chemokine...
Right. So I wonder if kine and chemokine is like the same root as kinetic.
I don't know.
I suspect that it might be.
I imagine that cytokine came first, and then chemokine came once they realized that some cytokines were chemotactic. I can find this out. I will.
That's
go
probably
do that.
true.
For the record, so we were actually sort of both correct here. So cytokine is cell movement, the basis of cytokine, and chemokine came after that when they realized that cytokines actually sometimes really do move cells. And so chemokine is just an amalgam of chemotactic cytokines, which is implied itself in the name cytokine. So basically the two things mean exactly the same thing, but they just needed to BS their way through it.
Well, okay then. Immunologists, we should do a whole episode on immunological vocabulary because it's kind of ridiculous sometimes.
Pretty absurd.
Anyway, we can do vocabulary and our obsession with defining cell types.
Right. And flow cytometry.
Well, that will definitely come into the cell type definitions. Anyway, so let's use T cells as an example. So T cells, as I mentioned, start as a hematopoietic stem cell. They develop into a pre-T cell and then a naive T cell. Right.
And then potentially a Rastafarian T cell. Go look at my Rastafarian T cell.
Right. Potentially a Rastafarian T cell. That's not a technical term. That's just a graphic that Matt made.
Yeah, I like it.
It's really good. It's pretty great. So as T cells are developing, first they move to the thymus, and when they leave the thymus, they have a particular localization program that tells them to find lymph nodes. And so they leave the thymus, they go and find a lymph node. But they're not stuck in that lymph node. They'll shut the program off briefly. If they can't find the antigen that they were looking for, they shut the program off, they leave the lymph node, they turn the program back on, they find another lymph node, and they keep on doing this. Sometimes they end up in the blood, but then they just go and try to find another lymph node. If a T cell gets activated, if a T cell is in a lymph node, sees the antigen that it's specific for being presented on the correct MHC, they get activated. They're going to do the things that we talked about in previous episodes where they blast, they make lots of copies of themselves. And then they're going to have a different location program. And this program is going to be dictated by the nature of the infection that they recognize. And more specifically, the type of lymph node or the location of the lymph node where they got activated. So, for instance, if a T cell is hanging out in the mesenteric lymph node, which is the lymph node that drains the intestine, it is going to get a signal that says you need to go to an intestinal type tissue because the infection that you are specific for probably came from the intestine because you were in an intestine draining lymph node. If instead it was in a lymph node that drains the skin, it's going to get a different program that says you need to go find inflammation somewhere in the skin. And T cells can't flow backwards up through the lymph to go straight back to the tissue that the antigen came from. They actually have to leave the lymph node.
They're not nearly as productive as salmon. They can't go upstream.
Right, they can't swim upstream, so they have to go.
They have to go.
They have
all
to
the way
leave
through
the exit door, flow back out through the lymphatics to the thoracic duct that I mentioned before, get into the blood, and then in the blood, they're going to be searching for inflammation. And they're going to be searching specifically for chemokines that are released during inflammation. So at sites in the blood where T cells could potentially migrate into tissues, there are a number of different molecules that are turned on that allow cells to sort of stick to the vessel wall because blood is moving through your vessels at an incredible speed.
Especially when you're a T cell and you're very, very small.
Right. I don't remember.
I think it was 70 body lengths. I remember the term 70 body lengths. I think it was a second or something like that. Like 70 body lengths of a T cell per second. I'm totally making up that number, but I'm pretty sure that I heard that once.
Yeah. Uli von Andrian, who teaches the lecture on T cell migration to us in our first year of graduate school, he has a lot of really impressive sounding numbers and images about the shear forces acting on T cells and how fast they're moving. Basically, imagine trying to take an off-ramp from a freeway when you're going like 500 miles an hour. It's just not going to happen. So,
or it could, and it would be catastrophic.
Right. So T cells, when they encounter a blood vessel that is near an inflamed tissue, that blood vessel is actually expressing molecules called adhesion molecules that the T cell can sort of stick to and start to roll. They actually physically roll along the edge of the blood vessel. And I think we're going to put up some movies of this happening on the post that's accompanying this this podcast.
In our freeway example, it'd be sort of like if you just slammed your car into the right barrier for a while, and that caused you to
and that
slow
friction
down.
sort of slowed you down a little bit. Right. Excellent metaphor.
Yeah.
And as they're rolling along the edge of this blood vessel wall, they're going to be looking for the presence of chemokines that they are programmed to find. So as I mentioned, in the lymph node, they're going to be programmed to look for specific types of tissues. Different types of tissues will express different sort of flavors of chemokines. So let's say you have that T cell from the gut, from the mesenteric lymph node in the gut, looking for a gut-like inflammation. It's flying through and it sees a blood vessel wall that is associated with the skin. It might stop and roll and look for that chemokine, but it's not looking specifically for the skin chemokine. It's looking for the gut chemokine. And so it's going to roll along the skin, the inflamed portion of the skin. It's not going to find its signal, and it's just going to pick up and keep going. So it's going to put the accelerator back on.
Jump back on.
Move
the
away
freeway
from the move away from the barrier.
Yeah, I actually sort of really like that metaphor.
I do as well.
All right.
It has a nice visual flavor.
Yeah, exactly.
So it's going to keep going through the blood until it finds the gut. And then it's going to stick to the blood vessel wall. It's going to start rolling. It's going to see the right chemokine, and it's going to then stick to that vessel wall. I actually screwed up. It's not looking for a different chemokine. It's the integrin is different.
Well, it needs the chemokine receptor. It needs the correct chemokine receptor still.
Yeah, but the chemokine receptor is standard, and it's the integrin that's different.
That's a long way back.
Yeah. Okay. I'm not sure exactly how to fix that.
I don't know either. I think that we should put an addendum. We're just going to cut this here for a second and say, I think both of us just used the word chemokine a lot. And in many cases, what we really meant was integrin. And all of the concepts still hold from what we just said. But...
Except we have used chemokine correctly, so that's going to be confusing. So T cell is looking for the right set of signals. And the analogy that Uli van Andrian, this guy that taught it to us, used is it's kind of like an area code. So if you want to call someone in San Diego, you're going to use the area code 619. If you want to call someone in Boston, you're going to use the area code 617. So even though two of those signals are the same, so basically there are a number of different signals that influence where T cells are finally going to migrate out of the blood and into the tissue. And they're different for different sorts of tissue. So the skin is going to be different than the gut, and a T cell that was educated in a lymph node that drains the gut is going to know to home towards the gut. The different molecules that influence this are chemokines and chemokine receptors, and another group of adhesion molecules called integrins. Integrins are what allow T cells to actually stick to the vessel wall. So as they're rolling along, they get a signal from a chemokine, it causes them to activate an integrin, and that integrin allows them to actually stick and halt on the blood vessel wall. As the blood is flowing through at rapid speed, T cell can actually stick to the vessel wall and then migrate through the barrier of the vessel wall into the tissue.
Right, and it's really that integrin interaction that provides the specificity there, right? So you have your T cell that you just mentioned. It got activated in a gut lymph node, so it knows that it was in the gut and it's looking for a gut-associated infection. It went all the way through the lymph. It drained into the blood. It went around. Maybe it saw a skin infection. It ignored it because it wasn't specific for a skin infection. It wanted a gut infection. Finally, it found a blood vessel that was close to the gut infection, and it started rolling. It slammed itself into the median like we were talking about. It took the exit ramp, and then when it took the exit ramp, it saw exactly what it was looking for in the form of the appropriate receptor for its integrin to bind, and that causes it to completely stop. It just stops in the middle of the highway, basically, is what Kevin is explaining.
No, it's on the off-ramp now, Matt.
Well, yeah,
it's
I
on
know,
the off-ramp.
But really, realistically, it's still in the highway.
No, it's still on the highway.
And so it just stops there and says, yep, I'm in the gut. And there's inflammation, and this is probably what I was activated for, and then it just goes right across. It just gets off the highway.
Right. And so to bring this back to the discussion we were having at the beginning about MS, T cells need to cross the blood-brain barrier, which is going to have a particular set of integrin signals, a particular set of signals that tells the T cell to leave the blood and go into the brain. And the fact that T cells need to do this in the brain is actually the basis for one of the more successful therapies for MS that we've developed. And that is a drug that's called natalizumab. The brand name of that is Tysabri. And what natalizumab does is it basically blocks the integrin that is specific for getting into the central nervous system. And so the great thing about this medication is that it's quite specific to the brain. This integrin that allows T cells to migrate into the brain is only found in the brain. And the reason that's important is, as we mentioned in our discussion of transplantation, when we want to suppress the immune system with drugs, we really don't have any drugs that specifically suppress the parts of the immune system that we want to suppress. Most of the time when we do immunosuppression, because of autoimmunity or because of transplantation, what we have to do is we have to suppress the entire immune system. And that's obviously a problem. The immune system is important for fighting off infection. And the great thing about natalizumab is that it specifically suppresses the immune system in the brain. And thankfully, there aren't a lot of infections in the brain that we have to worry that much about.
Right.
Actually,
there are a couple, though. There are a couple. And
yeah, there actually are a couple. And there's a known increase in the occurrence of this thing called progressive multifocal leukoencephalopathy, that's caused by a virus called JC virus. And most people don't have any problems with this virus. I mean, people can be infected with this virus and it doesn't cause any problems because usually the immune system is able to suppress it. Sometimes immunocompromised people can't, and people who are taking this medication for MS also can't because their immune system is prevented from entering their brain. And most people, I think, that have MS and most doctors that treat it would say that the low risk of getting this disease is well worth the relief of MS symptoms, because MS is pretty terrible. But it is immunosuppression. The good news is it's very, very specific immunosuppression, and it can be because we are specifically targeting a component of the immune system, this integrin, that is basically allowing cells to migrate out of the blood and into the brain.
So I plan on trying really hard not to say natalizumab too many times.
Okay.
That is my hope, because I won't be able to do it. So, but I think
you can say Tysabri instead.
Tysabri might work a little bit better. But yeah, we talked a lot in actually the first episode, the MHC transplantation episode, about just how devastating immunosuppression can be, and that you can control adaptive immunity at a very specific location, especially something like a brain, and something as devastating as MS is actually really cool.
So I hope you've learned a little bit about how cells move around and how that might influence a specific disease like multiple sclerosis. It has implications in a lot of other diseases as well. But next, we're going to talk about a specific paper that was published 2 years ago in 2012, and how the migration of cells can influence the etiology of multiple sclerosis.
Yeah, so again, your point from before, from sort of the intro section, where we can specifically inhibit T cells from getting access to the brain, is really interesting, and it's super important for human disease, and we have a lot of people benefiting from the development of that drug. One of the interesting questions, though, surrounding all of autoimmunity is really what is happening at the front end of autoimmunity. So we have the immune system. It's a big, powerful system. It influences almost your entire body on a really dynamic basis. But it also has a whole lot of checks that are built into that. We've talked about positive and negative selection just a little bit, and I'm sure that we'll talk about more. But basically that entire process is trying to make sure that you don't have any B and T cells that recognize your own body. And so it is reasonable to see a scenario where a T cell might accidentally go out into the periphery that recognizes, part of yourself. And then you have this additional system that we talked a little bit about last time, which is even if you have a T cell that recognizes something in your body that it is specific for. It also, in addition to seeing what it's specific for, also needs some sort of danger signal, right? So this is signal 2, we talked a little bit about, and again, we'll talk about that a lot more, I'm sure, going forward. But you have these different checks that make sure that a T cell that isn't supposed to get activated isn't activated. And so the breaking of those steps of tolerance is really what we're talking about when we're talking about the initiation of autoimmunity. Now, the problem in understanding exactly what happened, exactly what the first stages of autoimmunity. Welcome to Audiommunity, episode 4. The first stages in determining what exactly happened, we call it breaking tolerance. What broke tolerance at the initiation of autoimmunity is that when your immune system starts attacking your own body, you create all of these huge inflammatory danger signals, right? It's almost like you were infected by a pathogen. So a lot of the danger-associated molecular patterns that your adaptive system is going to pick up on and going to use in order to determine whether or not to respond, all of those are actually generated by the disease itself. And so once autoimmunity starts, it's sort of on a feed-forward loop. So you get the first T cell that breaks tolerance, and that leads to inflammation, and that leads to danger signals, and that leads to more T cells getting activated, and it just keeps going and going and going and going. And we'll talk about the actual mechanism of that, I'm sure, sometime in a more pure autoimmunity episode, but we call that actually epitope spreading, where at some point it doesn't have to even be clones of the initial T cell that are the problem. You start generating new T cells to different parts of your body, and it just sort of grows out of control. And this is one of the major problems with lupus and why lupus can attack so many organs. It's that your B cell population is basically just going nuts and it's responding to everything at that point. But the real question for a lot of immunologists is, how did that start to begin with?
Right, and it's difficult to find where it started because by the time anyone has symptoms of an autoimmune disorder, all of this epitope spreading that Matt has mentioned has already occurred. All of this inflammation, tons of T cells are active. By the time you can actually see a sclerotic lesion in the brain of someone with MS, or by the time you actually see the rashes that are associated with lupus, or by the time people are actually in pain from the antibodies in the joints if you have rheumatoid arthritis, by the time someone gets to see a doctor with an autoimmune disorder, all of this stuff has already happened, and it's difficult to go back to the beginning and say what was the cause in the beginning.
Right. And so really the only way to address this is to find a way to actually induce the disease and then track what happens during it, right? So you control the start of the disease and then you track the T cell populations. And so in this case, in the case of MS, we actually do have a really effective, pretty well representative experimental mouse model, but it actually also works in rats and rabbits and primates, and it's very similar to human MS. Call that EAE. So we call that EAE, which stands for Experimental Autoimmune Encephalomyelitis. So I'm going to call it EAE because I'm going to stumble all over that. But essentially what you're doing when you are inducing MS into a mouse model in this case, the paper that we'll be talking about talks about rats. But what you can do is you can take T cells that are specific for the brain of a rat and you can activate them. So you give them all of the normal danger signals that they would need. And then you take those T cells and you transfer them into a rat. And the T cells, they no longer require any additional activation signals. They find the brain, they cross the blood-brain barrier because they've been activated, and they start causing this MS-like disease. And that way you can track those initial T cells that you put into the rat at the beginning and say, we know that these T cells are the cause of the disease. What are the T cells actually doing? So at this point, I think we should probably say the title of the paper, which is T cells become licensed in the lung to enter the central nervous system. And the first author on that paper is Francesca Odeardi, and I apologize, as always, for any name mispronunciations here. last authors are Cassandra and Alexander Flugel. I believe Cassandra is actually Cassandra Flugel-Koch. So this paper basically centers around this idea where you can induce EAE in a very directed way just by introducing T cells that are specific for the brain and are activated. The question that they're asking, though, is what is the timing of all of this? What does the timing look like? So if I inject a T cell that's specific for the brain, that is, quote unquote, the start of MS, what is that T cell actually doing to gain access to the brain? And this, if we just sort of dive right in, I imagine that this whole paper probably started as a very investigatory, I want to know exactly at a really high resolution, hour by hour, what that T cell is doing. So they took these T cells that they had made so that they could track them. The way that they did that was they took T cells that glowed green, basically. So they had put GFP, green fluorescent protein, in those T cells. And again, those are T cells that are just specific for the rat brain. And they injected them into a rat. And then they waited, and they took a look at the rat, and they took a look at the spinal cords of the rat, which is one of the first infiltration sites when you start the MS process, the EAE process. And what they found was a bit surprising, actually. So they had injected all of those T cells, and it took 60 hours before you started to see those T cells actually getting into the spinal cord. And so there was a delay. And for, I guess, as a standardization, maybe it would seem like it would take 60 hours for the T cells to get there, to get to the spinal cord. And actually, that's not the case. If you injected a T cell that was specific and wanted to go to the spinal cord immediately, it would be there within minutes. So this is actually a very, very fast system. So the fact that it's waiting 60 hours before getting into the spinal cord suggested to them that maybe there's something else going on here that we're not quite seeing when we're inducing our EAE model.
The thing that I think is so amazing about this is that if you look at the graph in figure 1b, they're looking in the blood and in the spleen, and you can see that, so they're injecting these T cells into the blood. One of the great things about studying the immune system is that because of all of this homing stuff that we were talking about, you can put immune cells, even hematopoietic stem cells, you can just stick them right in the blood and they will go where they are supposed to go because they're really good at homing.
Have you ever heard that IP is the coward's IV?
No.
someone told me that once, and I really liked it. So
IP is intraperitoneal, which is the basically the organ sac in your torso, is the peritoneum.
Right, which eventually finds its way into your bloodstream. So there's a joke that, if you're injecting IP, you really should just be injecting IV. And, it's harder to hit a mouse vein than it is to hit an entire thoracic cavity, I guess.
Yeah.
Not thoracic, abdominal cavity. And the doctors stopped listening.
Yep. Anyway, so we're injecting these T cells into the blood of these mice, or of these rats, excuse me. And they have a time point that's 0.02 hours. So they're looking at the blood basically a minute after injection, and they find the T cells in the blood, which I suppose isn't super surprising.
Which they just injected,
which they just injected a minute earlier. But then when they look 6 minutes after injection, there's no longer any T cells in the blood. They're just gone. And they're not in the spleen, which is an organ where a lot of blood hangs out, and there's a lot of immune cells in the spleen.
It filters, it filters the blood. Yeah. And it's sort of it's another antigen.
It's like the
depot.
lymph node for the blood.
Yeah. I like
So, or maybe lymph nodes are the spleen.
No, because that would mean that B cell biologists have been right the whole time.
Anyway, so after 6 minutes, they inject these T cells. 6 minutes later, they cannot find these T cells that they injected in the blood or in the spleen. They've just vanished.
Yeah, and it's a testament to how quickly this homing can actually happen. And sort of along those lines, you inject into the blood, and they're not in the blood anymore. And you can't really find them in the spleen. And we know that they're not in the central nervous system because they haven't reached the spinal cord until 60 hours. So the obvious question is, where did these things go? And where did they go within a minute? right? Within 6 minutes, I guess, in this case. And so what they saw ultimately was that you get a really quick blip of T cells that you can find in the blood. If you look at 1B, basically what you see is that at 60 hours, 72 hours, which is when you first start seeing T cells in the spinal cord, you get a really big blip of T cells in the blood as well, which means that wherever those T cells went when you first injected them, they stayed there for 60 hours. Then they all rushed back into the blood and they immediately left the blood and they all went into the spinal cord, right? So there's this basically what they're doing is they're tracking this mass migration of T cells from the injection into some unknown area and then back into the blood and then back into the spinal cord. And so what they figured was, well, something must have happened. Maybe something happens to those T cells wherever they were in those first 60 hours. And whatever happened to them was the actual cause of them deciding to leave and go to the spinal cord, which I think is probably a pretty reasonable and testable idea. So what they do is they actually set up a shunt system between two rats. So this is kind of a cool experiment, actually. So you give rat 1 a whole bunch of T cells, and it's going to take 60 hours for those T cells to go from the injection into the central nervous system. And so at 48 hours, you take that rat and you hook it up through a blood circulation system with another rat who has not been given any T cells. And the idea is if the T cells themselves have actually acquired the ability to immediately jump into the blood and then into the central nervous system, what should happen is all of the T cells should erupt out of wherever they are in rat 1 at 48, 60 hours. They should circulate between both rats' bloodstreams because the bloodstreams are now connected. And
so I was going to ask this actually. This is a parabiosis? So they're actually...
This is not a parabiosis. I was actually glad that it wasn't a parabiosis because then I don't have to explain what parabiosis is and why we use it.
Okay, but the circulations are connected and can go from one to the other and back.
Yes, yeah. So there is basically just a shunting system that allows the blood to flow from one mouse to the other, but it is not a true parabiosis. Okay. But basically what they do is they hook up these two rats to the same blood system. And so and the second rat doesn't get any T cells at all. So when the T cells come out of wherever they're hiding at 48 hours from rat 1, those T cells will get shared between the bloodstreams of the two rats. And so if it's the T cells themselves that have picked up some ability to infect the central nervous system, they should be able to infect the central nervous system of rat 2 as well as rat 1 because they're going to be in that bloodstream. And that's actually exactly what they see. Basically, the observation comes down to the fact that it seems like wherever those T cells are in the first 48 hours following the initial injection, they pick up the ability to infect the central nervous system. And you can test that directly, and they do in this paper as well, but I think that that's probably the take-home message. So the question then becomes, of course, well, where are they, right? If they're not in the blood when you inject them into the blood, obviously, but they leave the blood, they're not in the spleen, they're not in the CNS, where are they? And this is like my favorite, it's one of my favorite method, overviews that I've ever seen in a paper. So how would you go about testing this? How would you go about finding where these T cells are? And so they decide to section a whole rat. So they take rats at different times during that first 48 hours and they fix them, sort of like an embalming process, basically, so that all the tissue is preserved. And then they literally cut sections of the entire rat, right? They image the entire rat section by section. It's a little gruesome, but I think I should actually explain what this is. Basically, what you have is something that's a lot like a deli slicer, right? Where you have a big ham and then you want smaller sections of that ham. And so you make a whole bunch of passes and you can image, just by microscopy, you can image each one of those sections and then look for the T cells that you injected. You can get section by section, you can take a look through and you can say, are the T cells in the liver in the first 48 hours? No. Are they in the intestine? Maybe some, they do see some. Are they in the heart? No. And then they took a look at the lungs. And it turns out that all the T cells that they were injecting, like virtually all of the T cells, were immediately infiltrating into the lungs, which is, sort of surprising, actually. And so they take a harder look at these T cells, and sure enough, over the first 72 hours, you just get this building of T cells which will eventually go into the central nervous system, accumulating in the lungs, and actually sort of infiltrating into the airways themselves of the lungs. They describe the airways of the lungs as these roads, almost, where they're going nuts, basically. They're increasing, they call it increases in velocity. Basically, the T cells are moving faster. All of these things are indications that the T cells are becoming active. And they're becoming active in a place that is really weird. Like, why would you expect a T cell that is specific for a protein in the brain to be in the lungs and getting activated? And to be honest, I don't actually know that. I don't know the answer to that, but
I wonder if it has something to do just with the sheer volume of capillaries in the lung. So obviously these T cells are going through the blood. If they're going to want to get out into some tissue, the place where there is the highest abundance of blood vessels in a tissue is in the lung, because the lung is obviously where oxygenation of the blood is happening. The lung is filled with capillaries so that all of the cells of your blood can pick up oxygen. And it's kind of interesting that the other place that they see them is in the gut, because the gut is also just perfused with capillaries. There's a lot of blood vessels that terminate in the gut as well. So I wonder if it's just a sheer numbers thing.
Yeah, and to be honest,
like maybe they would be in the skin. Maybe there are some in the skin, but
you're just not seeing
in terms of the numbers of capillaries, you don't see them there.
Well, so in this case, they do show that those T cells actually get into the airways themselves. So not only are they in the lungs, but they are crossing the barrier, the blood-lung barrier, for example. They're crossing the blood-lung barrier, and they're moving into your airways. And they are going crazy there. And I sort of agree. It actually worries me. This is something that worries me about this study, is that you have so much vasculature in the lungs that anything that you inject IV goes directly there. I mean, so there is some caveat there. That being said,
yeah,
I think it's probably
for an individual T cell, probably in real MS. So this is EAE. This is experimental. That's what the first E in EAE stands for. You probably don't have a bolus of myelin-specific T cells that starts actual MS, right? That was the main objection I had to this paper.
Sure.
It's a cool paper, but
it is a cool
that was the main
paper.
objection, is that it's I don't know how many conclusions we can draw that are actually important for the etiology of human MS,
right,
as opposed to a particular induction of EAE.
They do test non-brain specific T cells, and they show that even non-brain specific T cells do go to the lungs, and they do still seem to get activated. They were ova-specific T cells, ovalbumin, which is actually specific for an egg protein in chicken, and there are historical reasons why we use those T cells. But in any case, it was strange to me that they basically say that all T cells do this. It doesn't really matter what they see, but it's just these specific ones that find their way into the brain and cause this disease, which...
Yeah, that experiment makes me think this is an artifact,
right,
basically.
No, I know, but it was really confusing. I think what I took away from this paper in general was that mucosal surfaces are super confusing.
Yeah, that's definitely true.
That's
one thing that they did that I thought was interesting, is that they show that these T cells are able to cross back and forth across the barrier between the blood vessels in the lungs.
right.
And they showed that this is specific for T cells. So they took other types of cells, or they took T cells and they basically put them into the rats intratracheally. So they basically made them inhale the T cells, and the T cells were able to get into the lung from the lung side into the blood side.
Yeah.
But other immune cells were not able to do that. So crawling across an epithelial barrier is an active process, and the T cells were able to do that.
Right. No,
I don't know that naive cells would have been able to do that.
Yeah, that's the thing. Because they have to stimulate, like you mentioned or alluded to before, you have to stimulate these T cells before you inject them into the rat. You have to give them all those danger signals. And
right.
I just don't know what happens when you have a naive T cell or how relevant that is. In any case, I think what was interesting here, and it goes back to our T cell homing talking points a little bit earlier, was that it's interesting that they go to the lung. I don't exactly know what that means. They don't really talk about antigen presentation or anything like that in the lung, so I'm not clear about that. But what they do show really nicely is that while they are there, so these T cells have been injected in the blood, they go to the lung, and then something changes, and then they go to the central nervous system. And so they do a really good job of tracking exactly what is changing in those T cells over that time. And what they notice is a couple things. First of all, the expression of their chemokine receptors, and that is basically how these T cells know where to go, right? This is the program that gets you to move to the lymph node versus the program that tells you to go to the gut versus, all of those things. That is influenced by this chemokine signaling. They show very clearly that the T cells that go into the lung change their chemokine signature from one that looks like an unactivated sort of static T cell that would cause them to home to the lymph node to one that very rapidly will find its way to the central nervous system. And they do that a couple ways. One way, and I just wanted to introduce it briefly because I think we're going to come back to this a lot, is they do a gene array analysis, basically, or a transcriptome analysis. And briefly, you can think of DNA like a big book that has a blueprint for, every tool. Let's say you have a self-contained factory, right? And the factory can make all of the tools that it needs. And it's got one giant book. And that book tells you how to make all of those tools and when to make those tools, right? And so that's like your DNA. It's sort of like the blueprint that sits off in the corner. And then in order to make any of those things, you need to, fill out an order form or something like that. Say, we need 35 of these tools, right? And so that order form, that order comes in the form of RNA. And eventually those order forms get interpreted and the 35 tools are made. And so the end tool, the actual machinery itself, are the proteins of the cell. So the DNA is telling you how to make the protein. The RNA is telling you to make the protein now. And then the protein itself does the work, whatever work needs to be done on the other side. And so if you take a cell...
The central dogma, ladies and gentlemen.
Yeah, that is the central dogma of biology. So if you want to know what a cell is trying to do, it's not very useful to look at the DNA because the DNA is going to look the same whether you're looking at a T cell or a B cell, or it's going to look pretty much the same, right?
Or if you're looking at a skin cell.
Right, or a skin cell or a brain cell or anything, right? All of the DNA is going to look pretty similar because all human cells have the same blueprints. They have the same order book. But if you are a brain cell, you need very different tools than if you're a T cell. And so your orders are going to look a lot different in the brain than in your T cell, right? So if I have a T cell specific protein, a T cell specific tool that I need to make an order. I need to make RNA that requests that T cell specific tool. And so what you can do is, instead of comparing the DNA between the T cell and the brain cell, you can compare the RNA. And so you basically assemble a list of all of the active orders in that cell and say, okay, what's the T cell trying to do now? What's the brain cell trying to do right now?
Because in this case, it's two different T cells, but it's the T cells that are in the lung later about to leave for the brain, and the T cells that they originally injected.
Right, exactly. And so when you're looking at this system, if you take a look at the T cells that first get into the lung, they look a whole lot like they're trying to stay sort of naive, home to lymph nodes, all of those sorts of things. They've got all the hallmarks of a T cell that wants to just hang out in lymph nodes and not be a problem. But by the end of their time in the lungs, that has completely changed. They're producing different RNA, and as a result, they're producing different proteins, and those different proteins are going to cause them to move out of the lung and into the central nervous system. Now, specifically, what we're talking about here are chemokine receptors. We're talking about CCL19 and CCL21, which are the chemokine... we are looking at CCR7, which is the chemokine receptor that recognizes the lymph node-specific chemokines. versus CXCR3, which is going to recognize inflammatory stimuli. So that's the one that's going to direct these T cells to the central nervous system. So basically, they're downregulating CCR7, they're upregulating CXCR3, and they're basically going through this process where they're priming themselves to leave the lungs and move into the central nervous system, where they're going to actually establish the MS-like phenotype.
So the thing that confused me about this, and the next figure, which I guess we should just say, just to wrap up the paper, the experimental part of the paper is they basically show that, as Matt mentioned a little bit earlier, it's not just the brain-specific T cells that do this. Basically, any T cells that they put into the mite or into the rats are going to have this same behavior. And they characterize the behavior. They characterize this by just showing where these different T cells are going.
Right.
And the thing that confused me here is that if they're all getting the same activation phenotype in the lung, what is letting them get into the brain? We mentioned before about the blood-brain barrier. It's supposed to prevent the movement of T cells into the brain generally. And we also talked about the fact that you have to get a very specific signal to upregulate the correct integrin that allows you to get into the brain. That's the target of this natalizumab drug that works so well in MS. So how are they getting expression of that integrin? If all the T cells are behaving the same way, regardless of their TCR specificity, how are they getting the right migratory signals to get to the tissue where they're going to cause problems?
Yeah, a couple things confuse me, and I totally agree with you. The major thing that confuses me is why these T cells are getting central nervous system specific signaling in the lungs.
Exactly.
It is completely unclear to me why the lung is telling T cells to go to the brain, or the mediastinal lymph node is telling T cells to go to the brain. The mediastinal lymph node is the one that drains the lungs. Usually the mediastinal lymph node tells things to go to the lungs because that's where the danger probably is, right? So it is completely unclear to me why T cells would get a go to the central nervous system signal. As far as the non-specific T cells are concerned, it's possible that they're still going to the central nervous system, but they're just not causing any damage because once they get there, they don't see the correct things. So they go, but that doesn't get around to the first point, which is a much more important point, which is why T cells are going from the lungs to the brain in this scenario.
Exactly.
And I honestly, I don't know the answer to that. They don't talk about antigen presentation. They don't talk about influencing.
No, because you can imagine there might be myelinated neurons in the lung. I mean, the lung has nerves that innervate it. Some of them might be myelinated, so there might be some myelin present in the lung.
I wouldn't expect it as open antigen though, right? It should all be pretty well sequestered.
Yeah, but cells are dying. I mean, you're going to get some presentation of these things. I mean, I'm just trying to make a case for it. I don't actually believe it. It seems crazy to me. And yeah, it just doesn't make any sense. Like you said, why would cells going here? The conclusion I came to at the end of this paper is that this is a really cool phenomenon of something that happens in EAE.
Right.
But doesn't necessarily tell us anything about how an actual myelin-specific T cell in a human just before they get MS, how is that T cell getting activated? The etiology of the disease, like we said at the very beginning, figuring out the etiology of an autoimmune disorder is really hard. And I feel like this paper, there needs to be a follow-up to this paper that shows that, for instance, there was a particular chemokine receptor, a particular integrin or something that they identified.
Ninja 1.
Yeah.
It's NINJ1, and I just will call it
so like,
Ninja.
Would a ninja knockout mouse not get EAE?
I want so badly a ninja knockout mouse.
There actually is a gene called ninja, but it's not in humans, or it's not in mammals. Like we said last week, the fly guys and the plant guys get all the cool names.
Yeah, well, they generate the cool names. We get stuck with CD254.
Exactly.
Yeah. Oh well. Yeah.
And on that note, maybe we should wrap this up.
Yeah. No, I like you said, I think it's an interesting paper. It gets at this idea that we really do need to know what's happening at the beginning of autoimmunity, because that is ultimately what's going to help us prevent the initiation of autoimmunity, because it just gets out of control so quickly. But I am a bit concerned that this is a paper telling us what happens when you inject a giant number of pre-activated T cells into an animal.
Exactly.
So,
yeah, the title of the paper, as we said, T cells become licensed in the lung to enter the central nervous system. I feel like that title needs in EAE at the end of it.
Right.
Because I don't know that this...
in this system of EAE that we're using.
Right.
Right. Yeah, I don't know. I mean, and it's an interesting observation, and they do a good job of observing it. They do a good job of tracking what happens. It is a letter in nature. So,
yeah.
I mean, clearly people in the field felt that it was a really important advance in all of this, I guess, in the grand scheme of things. And who knows, maybe it'll turn out to be. Maybe in humans, what happens is you get an aberrant T cell, and then it gets activated, and then one T cell goes to the lung, and then it goes nuts, and then it goes into the brain, and that happens. But I would certainly like to see the follow-up studies.
Yeah. Well, as they say on Wait, Don't Tell Me, if any of that happens, we'll talk about it on Audiommunity.
And there it is.
So... yeah, I guess we'll wrap it up here. This has been Audiommunity from emunity.org. We are no longer on SoundCloud because SoundCloud is too damned expensive, but we are on iTunes now.
Yay!
You can also find the RSS feed on our website. Again, that's emunity.org. I'm on Google Plus and Twitter. We have a Facebook page, although I don't think we've actually put anything up on it yet. Facebook.com/Audiommunity. Eventually there'll be something there.
Our website is so much cooler than the Facebook page anyway, so...
That's true. I totally agree with that. On our website, you can find tons of really great graphics, most of which Matt has made because I have been working on my thesis and not able to do anything else except for record and edit the podcast. But all of those graphics, some really awesome animations, as well as other links can be found at emunity.org. That's EMMUNITY.org. We're supported in part by Harvard Medical School, and the music at the beginning and end of the podcast were composed by Rachel Reineck. Thank everyone for listening, and we'll see you next time.